Here you can see the glucose molecule is now getting converted to glycogen. That is glycogenesis. Whereas breakdown of glycogen to glucose is glycogenolysis. Okay. So you need to memorize all these steps. Okay. Glucose to 6-phosphate and so on. This is one box. Daily I am telling you, this is actually representing glucose. Between the glucose molecules, we have, what are these bonds? I said alpha 1,4 glycosidic linkages. At the branching point, we have alpha 1,6 glycosidic linkages. Okay, finally you will see the glycogen structure is formed. So through series of steps, memorize all those things. With enzyme structure also you should draw. So the glycogen molecule, whenever we require energy, it will be broken down. Okay, step by step, all glucose molecules will be liberated free. The mechanism of breaking glycogen molecule to individual glucose molecules is glycogenolysis. So you just draw these two cycles and frame the sentences in your own words. And he is also asking this regulation of this pathway. See, in the regulation mechanism, so here we have rate limiting enzymes. Rate limiting enzymes in the sense like glycogen phosphorylase. See, allosteric regulation. Here, whatever uh, uh, for glycogenolysis and glycogenesis, both are given in a nutshell. It is very good, in fact. When you just see glycogen phosphorylase, where do we get this enzyme? Let us check first. See, glycogen phosphorylase, as long as this enzyme is available, you will see the glycogen will be getting converted to glucose. Okay, so this glycogen phosphorylase activity is inhibited by the availability of glucose 6-phosphate, ATP. Already lot of glucose is there in the liver. These three factors will stop the activity of glycogen phosphorylase, so that glycogen will not be converted to glucose. Okay, this glycogen phosphorylase is rate limiting enzyme. Okay, glucose 6-phosphate, ATP and liver glucose will stop the activity of glycogen phosphorylase, whereas the skeletal muscle, if it has more calcium, that means muscle is working fast, more glucose is required, and AMP. So in these conditions you will see like there is more requirement of glucose. Hence, glycogen phosphorylase will get a positive information, a positive feedback. Hence, it will work more to break more glycogen to glucose. Okay. So, with the help of this flowchart, you can write the regulation. Now, glycogen synthase. So, this is the rate limiting enzyme here. Where do we have this glycogen synthase? See here. So during the process of glycogenesis, so this step where you see the UDP glucose is converted to glycogen primer, and this glycogen primer is again the chain is elongated, we require glycogen synthase. So this glycogen synthase is rate limiting enzyme for glycogenesis. Okay. So glucose 6-phosphate, as long as it is available, it will promote the activity of glycogen synthase. Okay. So this glycogen synthase continuously converts the glycogen to glucose 1-phosphate. So this is regulation. Okay. So whatever is shown in negative, that means these factors are stopping these enzymes. Whatever are shown in blue color, these enzymes are actually promoting. These factors are promoting these enzymes. Okay. You can frame these things into sentences. Because vitamin A is discussed many times, we will do beta oxidation of fatty acid. And as an exchange for this essay, let us see which page number it is. Beta oxidation 285. I think I gave beta oxidation also once we did. Okay, but still we will revise because it's an essay question. See here. Beta oxidation of fatty acid. What does it mean? See, the fatty acid, for example, palmitic acid is now being broken down. Okay, the fatty acid like palmitic acid is completely being broken down to form the end product acetyl CoA. Okay, So this is palmitic acid, and it will undergo a cycle called as beta oxidation at least 8 times. 16 carbon atoms will be broken down to finally 2 carbon compound, that is acetyl CoA. It takes at least 7 times cycle to break the palmitic acid to final acetyl CoA to give the required ATP. The definition is breakdown of a fatty acid like palmitic acid to produce energy is nothing but beta oxidation. Okay, beta oxidation. So beta oxidation, for this beta oxidation cycle takes place in mitochondria. Only first step takes place in cytosol. Okay, this fatty acid is, for example, palmitic acid. It is converted to acyl CoA. Further enzymes are not available in cytoplasm. Hence, acyl CoA is now transported to mitochondria, where all enzymes are available. Now, for example, there is a carnitine transport mechanism which you are supposed to draw like this. Okay, so acyl CoA is now getting transported from cytoplasm. It is now transported to mitochondrial matrix. For the purpose of transport, the vehicle what we use is carnitine. So acyl CoA combines with carnitine to form acyl carnitine. So this acyl carnitine is now moving from the cytoplasm to the mitochondria. So once acyl carnitine is entering into the mitochondria, it is again splitting to acyl CoA and carnitine. Carnitine job is over, hence carnitine comes back to cytosol and acyl CoA enters into the mitochondrial matrix. You can see this is acyl CoA. So it has successfully entered into mitochondrial matrix. So from acyl CoA, you will see trans-enoyl CoA. Okay, the enzyme we have here, acyl CoA dehydrogenase. Here one FAD is converted to FADH2, which will go into electron transport chain. According to old edition, from one FAD we have 2 ATPs. From this trans-enoyl CoA, we have beta hydroxyacyl CoA. Then from here we have beta ketoacyl CoA. From here we have acyl CoA and acetyl CoA. Okay. So there is a code to remember this cycle called as FATCA. FATHKA. You can write on the your page like FATCA. If you write, it becomes easy. FATHKA. You can remember the steps easily. Fatty acid, acyl CoA, trans-enoyl CoA, beta hydroxyacyl CoA, ketoacyl CoA, acyl CoA, and acetyl CoA. FATCA. FATHKA. Make a note of this. Okay. So, but this cycle, see how many carbon atoms palmitic acid has? 16 carbon atoms it has. So, one single cycle will not be sufficient to release all 16 carbon atoms at a time. In each cycle, 2 carbons are released, you see, CH3C. So 2 carbon atoms are getting released, and this acetyl CoA is entering into citric acid cycle to give ATP energy. So like that, this cycle will repeat 7 times. Okay, 7 times NADs and FADs involved here are around 7 FADs and 7 NADs. We know from 1 NAD, according to old books, it is 3 ATPs, 1 FAD, 2 ATPs. Grand total, if you do, we will get 35 ATPs. From citric acid cycle, also acetyl CoA goes 8 times. Okay, so 8 times each cycle will give you 12 ATPs, and 8 times the cycle repeats. Hence, you multiply 12 into 8, it comes to 96. So energetics are asked, no? You are supposed to write energetics in this way. In new book, whatever table has been given, you just convert the like figures, because now I remember NAD, how much does it give? You can share here if you want. Okay, maybe 2.5 and 3.5, something like that. So 7 FADs, 14 ATPs, 7 NADs, 21. Old terminology, 3 into 7, 21. 8 acetyl CoA, 96. Do the grand total. You will see one palmitic acid will give 131 ATPs, but initially it has utilized 2 ATPs. Hence minus 2, it comes to 129. Here ATPs were utilized, which you do as minus 2. Okay, hence the net gain of ATP in this beta oxidation energetics is 129 ATPs. Please convert it into according to new terminology of latest books. Ketogenesis, this also I already discussed. I hope all were there. Ketone body formation, ketogenesis, ketolysis. If anybody wants, write it in the chat box. I'm skipping this. Porphyrias also I have told. Competitive inhibition. These two answers I'm skipping better. If you want, write in chat box. Competitive inhibition I'm taking you. Or you can message me personally if you don't want to reveal your identity also is fine. I don't mind repeating. Please don't hesitate. So competitive inhibition. See here, basically the enzyme has an active site where substrate will come and bind. Okay, so when substrate comes and binds, we will get the product. But unfortunately, suppose if substrate and inhibitor are same to look at, then what happens if competitive inhibitor comes and binds, we will not get required product. Yesterday, we have seen one last step, no? Hypoxanthine to xanthine, xanthine to uric acid when we were doing purine metabolism. There the inhibitor I said allopurinol. Okay, so suppose that instead of this hypoxanthine and xanthine, suppose if allopurinol comes and binds, you will see uric acid will not be produced. So generally, competitive inhibitors we use as drugs to treat certain diseases like gout. So that type of inhibition is called competitive inhibition. Here also examples are given. Under that allopurinol only is given. I remember. See, this is so many examples we have for competitive inhibitors. So substrate is hypoxanthine and xanthine. In the place of substrate, if allopurinol comes and binds, xanthine oxidase will not be able to produce uric acid. Hence, we can stop the uric acid production to treat gout. Similarly, in the place of catecholamines, we can use ephedrine, amphetamine. Okay, if we want to treat catecholamine levels, if they are high in the blood, we can use these drugs. Okay, like that, many competitive inhibitors are used in the form of drugs to treat various diseases. Okay, you can choose 3 to 4 examples. Okay, you can draw this diagram. First is enough because it is competitive inhibition, and you can write this explanation. The inhibitor closely resembles the real substrate. Basically, if substrate binds, we get the product. But suppose inhibitor comes and binds, we do not get the product. Okay, till here you can write enough. Okay, and give 2 to 3 examples. So, question, just a minute. Keep reading this enzyme inhibition. I'll come get back to you. So that is all about this. Then we will talk about next Kwashiorkor marasmus. Kwashiorkor marasmus is nutrition chapter. Let me check the page number. What is Kwashiorkor and what is marasmus? Do you remember? See, here we are. See, basically, these are protein energy malnutrition disorders. Okay, what happens in Kwashiorkor is, see, the baby is receiving only carbohydrate diet. So the carbohydrate in the form of rice, gruel and all, and the baby is not receiving other nutrients like proteins, vitamins and minerals. Hence, the baby will be obese and there will be edema. Why there will be edema? Because proteins are not there in the diet. Hence, plasma proteins are not synthesized. So when plasma proteins are not synthesized, what happens? Plasma will leak into tissue spaces and there will be edema. The baby will be chubby here with round moon face, web neck, pod belly. Okay, so that type of thing you will see in case of Kwashiorkor. Whereas in case of marasmus, all skinny child, you will see through skin all bones are seen. At least this baby has rice to eat, though other nutrients are missing. But here, unfortunately, marasmus case, neither proteins, nor fats, nor minerals, nothing is there to eat. Severe starvation. So in such case, you will see the baby becomes very much skinny. So that condition is marasmus. Okay. So the age, few differences are given, but I just go through once. It's very simple and easy. Then we will go with galactosemia. Name essential fatty acids. Linoleic acid, linolenic acid and arachidonic acid. These are essential fatty acids. What is the meaning of essential fatty acids?
Emulsification of.
They cannot be synthesized by the body, hence they should be compulsorily taken in the diet. They are called as essential fatty acids. Okay. What is galactosemia? 270. Emia means always, I tell you, emia. Whenever blood levels are rising, we use the word emia, no? Galactose levels rising in blood is galactosemia. See. Galactosemia, disorders of galactose metabolism. Under this we get galactosemia. So galactosemia is due to deficiency of enzyme galactose 1-phosphate uridyl transferase. You will see galactose is not being metabolized in the body and galactose will accumulate in the blood and it will rise in the blood. That condition is called as galactosemia. Increased galactose levels in the blood is galactosemia. Okay, it's a rare congenital disease in infants. Okay, so galactose metabolism, you can just draw one. Here you can draw this step better. Galactose 1-phosphate is not getting converted to glucose 1-phosphate because of the deficiency of this enzyme. Galactose 1-phosphate uridyl transferase. That is what is galactosemia. Okay. What are isomerases? This again comes in enzymes only. Actually, it is 5 marks question. I will discuss like 5 marks only. Physically, they are not similar enzymes to look at, but they catalyze the same reaction. They are called as isomerases. Okay, I'll give you few examples. You will remember very easily. See, isomerases or isoenzymes? What is? Oh, isomerase. Okay. Isomerases, isoenzymes are different. Okay, in isomerases, where do you get beta? In carbohydrate chemistry, we have two isomerases, no? D-galactose, L-galactose, dextroform and levoform. They are generally called as isomerases. Okay, we will see. See, isomerases, enzymes involved in isomerization reaction. But he is asking two examples, no? So let us see here. Yeah, here examples are also given. Triose phosphate isomerase, retinol isomerase, phosphohexose isomerase. Just give example and give the definition enough for this answer. Sickle cell hemoglobin I discussed already. Sources and functions of folic acid. This you will find in vitamins. So, fructosuria and hereditary, this we will go. First we will see vitamins. What is the active form of folic acid? THF, tetrahydrofolate. It is seen folic acid is found only in green leafy vegetables. Folium means leaf. We do not find this vitamin in non-vegetarian sources. Okay, folic acid. And folic acid is very important for one carbon metabolism. You see folic, folate. One carbon metabolism, where did we see transmethylation also? Transmethylation is also one carbon metabolism. One carbon transfer. Yes. So I told you, folic acid is, folium means green leaf and it is found abundantly in green leafy vegetables. So under this he is asking us sources, right? And functions. So sources, you have to list out the sources. The sources specially it's given, see here. Green leafy vegetables, whole grain cereals, liver, kidney, yeast, eggs, milk is of course a poor source. Okay. Then what are the functions? I told you it helps in one carbon metabolism, transfer of one carbon wherever is required. You can give many examples where what is the active form of folic acid? I said tetrahydrofolate, THF. THF is active form of folate which helps in one carbon transfer. So one carbon transfer can be in the form of all these forms. N5 formyl tetrahydrofolate, N10 formyl tetrahydrofolates. Many examples are given. You can give those. That is more than enough for 2 marks. Then we will go with last question, fructosuria. What is fructosuria? Fructose is excreted in the urine. That means fructose metabolism is not taking place properly. Okay, we'll see this page number and I'll take you to that. It will be in carbohydrate metabolism, fructosuria. So we do not have enzymes which can metabolize fructose. Hence you will see it's excretion in the urine is increased and the baby cannot tolerate fructose. That is fructose intolerance and fructosuria. And here you should know the enzyme defect, that is more important. Which enzyme is defective? We will see that. Yeah. 280 page. हाँ डालो डालो जल्दी डालो. Aldolase B will be defective beta, because of which you will see fructosuria. Here you will have the explanation. Hereditary fructose intolerance. This is due to the absence of deficiency of enzyme aldolase B. Okay. So because of which fructose will not be digested and you will see the fructose levels will increase in blood. Okay, severe hypoglycemia, vomiting, liver failure, jaundice. Okay, so in such cases, we should see that baby is not given a diet containing fructose. Strict diet which is free in fructose formula should be given to such babies. Okay, beta. So that finishes. We will stop here, and you can join anatomy tomorrow same time. Okay. Study well.